Thank you, and yeah, thank the organizers more broadly for organizing this meeting and for including me, and yeah, having got this really fantastic location. All right, so I'm going to talk about ongoing efforts to connect molecular and cellular scale behaviors in microtubule self-organization, or an alternative title is why and how my group is starting to use techniques related to machine learning. You could argue whether it's really machine learning or not. Maybe we will argue about it. Okay, so in this is going to be a bit of an unusual talk in the sense that I'm not going to give you a polished scientific story. It's not really going to be polished, and it's not really going to be a scientific story. But nonetheless, hopefully you find it interesting. This is really going to be about, kind of how my lab is starting to get into a new area, why we're doing that, and what we've been doing. And like I said, hopefully you find it interesting, and hopefully it, I don't know, sparks conversations. All right. Okay. So yeah. So like I'm going to give, like I said, background and motivation, and then I'll really talk about, again, this new area, which is, and what we've done so far is really just technique development, which is really just stuff which is in progress. And like I said, as this is not very polished, I really hope that if people have questions, I really encourage you to shout out or raise your hands, and don't wait till the end. All right. So first, motivation and background. All right. So just starting very big picture. So what I'm interested in, broadly speaking, is biological self-organization. I'd like to have a quantitative understanding of biological processes, including how molecules self-organize into subcellular structures, how cells self-organize into tissues and organisms, and how these are perturbed in disease and change over evolution. Obviously these are very difficult problems, which can be studied in many different ways. The way that I find most satisfying personally is to try to gain insight into these issues by doing quantitative experiments that can be combined with theory and simulation. All right, so one of the things which we've spent a lot of time focusing on is particularly self-organization of microtubules in molecular motors. So as Steffi was just telling us about, so the spindle made up of microtubules in molecular motors, and they self-organize to give rise to that. And then also, obviously, microtubules and molecular motors are very important in many other contexts, including in neurons. So axons and dendrites are filled with bundles of microtubules. And then obviously the assembly of microtubules, their self-organization is very different in spindles and neurons, but they contain very similar components. Even many of the same motors and cross-linking proteins are important in both of these systems. So mostly my lab has studied these kind of things on the cellular scale, really looking at cellular scale phenomena. And so asking, for example, trying to look at spindle formation, how do we understand that? Spindle positioning, chromosome segregation, stuff really at the whole scale of the cell. And more recently, we're also starting to do look at cellular scale phenomena in neurons, including trying to understand growth of axons and dendrites, internal trafficking, and internal organization. All right, and not only are the questions we've mostly been asking kind of focused on the cellular scale, but the techniques that we use also really focus on the cellular scale. And so just to give you a sense of what I mean by that is, so for example, we try to learn about forces involved in spindle position. And other things, we use laser ablation, so you can cut the spindle and see how things move around, and then try to infer what forces are present. Or we can use magnetic tweezers, so put a magnetic bead in the spindle and exert forces and see how it moves. And so this is interesting, but again, you have to think this really is telling you stuff on the cellular scale, because you have a micron-sized bead, which is plowing through the spindle. Optical microscopy, obviously, tells you stuff, micron scale. And then to interpret this, we often use models, often continuum models, to try to understand this data. So for example, we've developed and tested continuum models of the spindle. So how to collections of microtubules work together to give rise to the spindle. And again, because these are continuum models, I mean, they may tell you about the concentration of microtubules or motors, but they're really all, and how those vary with space and time. But they're really all formulated on this very, very large length scale. Similarly, again, we do a bunch of photoactivation and photoconversion type experiments, and I'm just honestly showing you this data because I think it's cool. But this is a neuron, with an axon, which is, hundreds of microns long, in a graduate student in Lab Mara, photoactivated tubulin or microtubules, at many, many, many different spots along the axon. And then she can track to see how these spots move and then try to infer what microtubules are doing. And so, for example, she can look as a function of distance along the axon, what's the speed that microtubules are moving? And then she sees that as you get farther and farther away, the speed grows more and more. Approximately linear, which would be consistent with this, perhaps, this is very preliminary, this entire bundle of microtubules that fill up this massive axon, just continually extending everywhere along its length, right? And so it's kind of every part extending, so the whole bundle would be tensile. Anyways, but the point is that these measurements and this type of analysis, and even thinking about it in this way, is all at this very kind of like, macroscopic large length scale. All right, but, oh yeah. And that's great, very interesting, but, it's also very interesting to ask, how do these cellular scale phenomena arise from the actual molecular behaviors? Right? Not just thinking of formulating stuff at the cellular scale, but how does it arise from the actual molecules that make stuff? And so, I mean, like one way that we tried to get at that, and other people do analogous thing, is that maybe you do measurements at the cellular scale and you perturb specific molecules and you see what happens, but still, that's really still focusing at the cellular scale. Ultimately, we really would like to connect to, really the details of what's happening, in the how the actual molecules are really working. Oh yeah, and there's many reasons why we would want to do this. So first of all, it's actually interesting. I just want to understand, like really how do these collections of molecules, in some really true multiscale sense, give rise to these large scale behaviors. And ultimately, we really like to be able to predict, or at least understand, how molecular perturbations impact these cellular behaviors. And so really asking, if you have a genetic variant that leads to this, modification of this amino acid that has this, effect on, the free energy of binding of this protein to that protein, how does that impact stuff at this scale? All right. Now, that is very, very challenging. And it's very challenging for multiple reasons. One of the reasons it's very challenging is that so we have, very good techniques for measuring these cellular scale behaviors, like I was saying, optical microscopy, laser ablation, whatever, or an understanding of stuff at this large scale behavior, such as continuum theories. And of course, there's also very good experimental techniques to tell us about stuff at the molecular scale, so all of structural biology, right? And so X-ray scattering, NMR, molecular dynamic simulation, etc. But bridging the molecular scale to the cellular scale really requires understanding what's happening at these kind of mesoscales in between. This kind of scales of like tens of nanometers, which are large compared to individual molecules, but small compared to anything you can see in an optical microscope. And those are just very difficult to study experimentally. They're very difficult to study, but they really are key to bridging this molecular and cellular scale in a really serious way. All right, and so I mean, I think this statement is true in general, that this is interesting, important, and difficult. But thinking about microtubules and molecular motors in particular, like just to give a sense of what I'm talking about, understanding this mesoscale phenomena. So for example, all right, so microtubules cross-link molecular motors and slide them apart. Very important, again, for spindle self-organization, for neurons. But, I and other people, draw cartoons like this all the time. And the reason we draw cartoons like this all the time is because all we have is cartoons. And what's hard is that if you were to try to construct a theory of how the interactions between microtubules, molecular motors, lead to kind of sliding of microtubules and the generating of forces, it really depends on a lot of details of what's really happening there. How precisely are the motors organized and arranged between the microtubules? And we just don't know those details. I mean, so just as one example, like if you have a bundle of microtubules cross-linked by molecular motors, we always are thinking that the motors are really all between the microtubules. But, is that true, right? They could be on the outside of the bundle. They could be, like maybe if you have two microtubules aligned with each other, maybe only motors that kind of on one protofilament of the microtubule can cross-link to another particular protofilament, or maybe they can, do all sorts of angles and orientations, and that really has large implications for what's going on. Similarly, with other types of phenomena, even just thinking about motors on individual microtubules, so stuff like traffic jams and end accumulation, that depends on really details, or can, in theory, depend on really details of how motors interact with each other, and just the geometry of motors and microtubule interactions. And again, a lot of that we don't understand kind of in situ how those are happening. All right. So what the kind of premise of my talk and also my thought is that recent advances in cryo-electron microscopy really present new opportunities for studying these kind of mesoscales, both the kind of biophysics and physics of what's happening there. So that's what I'll tell you about. All right, so cryo-EM, so obviously everything I'm saying, kind of a lot of this really builds off of stuff which Steffi was talking about. But I'll just, I don't know, repeat or just say some more stuff. Okay, so just so we're on the same page, so cryo-EM is electron microscopy at cryogenic temperatures. So when you are doing cryo-EM, your sample is made of, your proteins are in vitreous ice, so amorphous solid water, not plastic. That means that the proteins are as in close to native state as possible. And the contrast really comes from the biomolecules themselves, not heavy metal stains, as would happen in plastic samples, which means that you can get as high resolution as possible. And with cryo-EM, it's very widely used to determine the structure of molecules in vitro in isolation, and also used to study molecules in cells. All right. And then as Steffi was saying, if you're doing work in cells, you need some way to have a thin section of your sample, which can be done by focused ion beam milling or other methods. All right, so with so the way conventional cryo-EM works is as follows, and this is like now incredibly well established. So you take your sample, your proteins in solution, you freeze it. You acquire cryo-EM images, which would look something like this. And these images are incredibly high resolution, but low signal to noise. Very, very low signal to noise. And therefore, what people do is that they have sophisticated algorithms to essentially align properly align all these noisy images of individual molecules in their sample, and then average them together in the right way. And then they obtain a high resolution structure. So that's conventional cryo-EM, which is, like I said, super well established. One thing which I'll talk about if people are, well, I'll just say, didn't have slides on this, but all right. So there's also kind of two ways of doing stuff. There's cryo-electron tomography, which is what Steffi was talking about. And then there's single particle cryo-EM, which is what's kind of depicted here. The difference is as follows, is that in tomography, what one does is one tilts the sample and acquires multiple images and then uses that to get a 3D reconstruction of the sample. The advantage of cryo-electron tomography is you really do get a 3D reconstruction. The disadvantage is that it's lower resolution. And it's lower resolution because you have to essentially divide up your electron dosage, because electrons are always going to kind of damage your sample. So you have to divide your electron dosage between all the different tilts. And then also you always have just some error in kind of like, mixing those tilts together to do a 3D reconstruction. So there's cryo-electron tomography, and then there's, single particle cryo-EM. Anyways, okay, so those are kind of conventional. But quite recently, so cryo-EM has been, there's just continual incredible advances in cryo-EM. And because of these, there's really been some really cutting edge applications of cryo-EM. And the way I think about it is that these cutting edge applications really leverage the single molecule nature of cryo-EM images, because these are really, you're really taking actual, images of individual molecules. All right, and you can kind of divide these into two different categories. So one is that instead of determining the average, structure of your protein, you can try to determine the probability distribution of different molecular conformations your molecule can be in. All right, and so then instead of because in a typical cryo-EM experiment where people are trying to get the high resolution structure, they may take images of literally like a million molecules, but then they end up throwing out maybe 90% of those images because they don't average together well. And the reason and there are multiple reasons why they don't average together well. One of the main ones is that the molecules are not just in one conformation; they're in multiple conformations, right? And so then, if one applies these techniques, and if they work, which is maybe a big if, then it's it's really amazing, because if you can really determine the probability your molecules is in different conformations, well, if your molecule were in equilibrium and it was just fluctuating around due to thermal fluctuation, this would allow you to essentially directly measure the free energy landscape of your molecule. If your molecule is not in, and then also get binding interaction energies essentially directly by observation. And then if you're not in equilibrium, like let's say if you have a motor walking on a microtubule, again, you still determine if you could determine the probability that you were in any given conformation, this is essentially, the molecular distributions which enter kinetic theories of non-equilibrium processes. So that it would be, fantastic. Okay, another thing, so that's determining, probability distributions. Another thing is inferring the localization and conformation of individual molecules in your sample. Right, and here there's different ways that you can do this. So one approach is to kind of determine molecular localizations and conformations essentially by reconstruction, right? And so, for example, this is basically what Will did in the work that Steffi was talking about, where what he does is that so you have a bunch of microtubules in your sample, and like I said before, in order to get high resolution structures, what you have to do is average, like kind of different regions together. And when you're, so then he selects a bunch of regions of the microtubule, throws them into a reconstruction algorithm. That reconstruction algorithm takes each part and tries to best align them. And then when it aligns them, it can determine, a high resolution reconstruction. But then you can figure out essentially for every kind of region which went into the reconstruction, what was its original orientation, relative to each other. And then that allows you to do that allows you to determine, for example, the polarity of microtubules. So that's, you could say as, localization or orientation by reconstruction. Another approach is to leverage all the, information that we have in structural biology. So for example, if you have a cryo image of a bacterial cell, and this is work by Brownman Lucas and Nico Grigoriev, what they do is they say, all right, we know the structure of the ribosome, right? Other people have great structural biology on that. And then you can just say, all right, given that I know the structure of the ribosome, let me search this image and ask, search all possible, locations, all possible orientations, and then try to figure out where the ribosomes are. Right, so that's leveraging the structural biology which other people have done. All right, and then with that you can try to bridge molecular and structural biology to cell biology, and obviously, or perhaps Oh, I don't know if it's obvious, but this would also provide input to develop and test real multiscale models of how things are happening. All right, so these types of approaches are really undergoing continuous development from many research groups, and they're really cutting edge, work, which is continually being improved. And so there's always a question, people are both developing better algorithms to try to do them, and then there's all sorts of different approaches, like I was kind of indicating, that people use, and then there's all different ways that, because one concern, right, is that you run some algorithm and it says here are the ribosomes, but like, how do you know it's right? Right? It'll give you some answer, but like, all right, okay, into some cases, people have developed very good arguments to show that they can get things right. And so, for example, what Brownwin and Nico did, which I think is very, very clever, is that they used a template which was missing part of the molecule, right? So a template with a hole in it, and then they, identify where they think all the molecules are, and then they use the location and orientation that they infer to try to do a 3D reconstruction of the missing part of the template. And then they can get a high resolution reconstruction, and that would only work if they actually did a good job of matching stuff, right? And so one can do stuff like that. All right, but anyways, continual development for many groups. All right, but nonetheless, I think these are really phenomenal techniques. And so then what, in my mind, or what I think would be amazing is to use cryo-EM to determine the localization, conformation, probability distribution of molecules in situ at these mesoscales, and use that to help bridge molecular and cellular scale. All right, and then that would inform and test both kinetic theories and kind of multiscale models, those type of things. All right, so the ultimate goal is, of course, to apply these approaches in a cellular context. And so you heard about the amazing work which Steffi and Will are doing in spindles. I'm also collaborating with Young at NYSPC, who's at NYSPC in Flatiron, who's doing large-scale who's doing large-scale cryo-EM reconstructions of entire axons in dendrites. And so that'll be completely amazing, and try to apply such approaches to those types of data sets. But computational and modeling challenges still currently limit the information that can be extracted from such cryo-EM data. Like I said, there's still a lot of development that needs to be done, to apply these, let me rephrase that. So certainly, like the results already that Will and other people are getting are just phenomenal, but there's clearly much more information contained in that raw data, and extracting that is still a challenge. All right. Yes.
One challenge that I had when working with these types of data is how do you go from static data to dynamics? Do you have any thoughts on that?
Oh yeah. No. So it's definitely important point. I think ultimately one will need to combine different data sets that, so for example, if you have a system, the simplest case would be, let's say you have a system which is in steady state. And on the one hand, you can measure the distribution of different, things by, for example, cryo-EM. On the other hand, you could measure at the light microscopy level how things move around, and their dynamics. And then it's a question of trying to combine them, but integrating them with some, I would propose some type of mechanistic model of what you think is happening, basically.
I'm curious to learn a little bit more about your neuronal preparation. Is this a primary culture system? Are you looking at, as you probably well know, there's thousands of different kinds of neurons.
100%. Yeah. No, 100%. And so like the main thing that we've been working on so far is primary mouse hippocampal neurons. And so, one gets them from a young mouse, and actually, I think it's kind of an amazing system because so you get from a young mouse, and when you get them from the young mouse, as you probably know, but other people may not, you actually damage them, and you end up with just little round cells, but they're still neurons. And if you throw them down on a glass slide, and then over the course of 2 weeks, they regrow their axons and dendrites, and they become, end up with a morphology which is quite similar to what they were in vivo. We're also starting to do some stuff with stem cells as well, but right now, the mass hippocampal primary neurons are the main system. All right. Okay. All right. So now I'll tell you about technique development we have done and are doing to try to address some of these challenges. All right. So a graduate student in my group, Michael O'Brien, created CryoJAX, which is what I'll tell you about now, which is a flexible image simulation library which leverages advanced scientific computing tools. And I think this is really very helpful for a variety. It will help really overcome many challenges in cryo-EM data analysis. Well, this is really Michael's baby. Now there are a wide variety of additional developers for CryoJAX at different institutions, and this is now currently being used by multiple groups in multiple projects. And the idea behind CryoJAX is it's really a modeling framework for cryo-EM image formation, which can link to advanced scientific computing tools. So CryoJAX is a flexible forward model for image simulation. And why that's interesting and important is that if you have a flexible forward model for image simulation, you can use that. It really helps doing Bayesian analysis of the resulting data. It's differentiable, and if you're not familiar with what that means, I'll explain that in a moment. And this quite easily integrates with modern tools for statistical inference and machine learning. The kind of power, or one of the main reasons that all this is so powerful, is that it's based on JAX. Which have people heard of JAX? Some people. Okay, so JAX is a Python library developed by Google for high performance computing and machine learning. All right, so JAX, so I'll tell you a little bit more about that. So this is, like I said, a Python library developed by Google. It has many incredibly powerful features. So it has just-in-time or JIT compilation, which means that code is compiled during execution to optimize performance. It's very convenient and very efficient for parallelization across GPUs and CPUs. And it enables or has kind of built-in automatic differentiation. All right, so for people who aren't familiar with automatic differentiation, this is an incredibly powerful tool, and it allows you to take exact derivatives with a computational cost that is independent of the number of parameters. And so what does that kind of mean? Okay. The reason that's important, or one of the reasons why that's useful and important, is that many optimization algorithms use derivatives of some loss function with respect to parameters to optimize something, right? And so, like, you're trying to optimize, the fit of this to that. You invent some loss function. You want to know how does this loss function improve or disimprove or get worse as you pay parameters, and this allows you to kind of compute that, very well. And automatic differentiation allows optimization of millions of parameters in neural networks, right? That's how they can train these things, right? And so, we care about matching stuff to data. We don't have millions of parameters, and people can do it with million parameters. That sounds, kind of promising, or at least potentially useful. All right. And so, and I think, this presents new opportunities in physical modeling and scientific computing. All right, so then, furthermore, CryoJAX is a very flexible image simulation library, and so So for example, there's really flexible representation of volumes or structures. So for example, you can represent them as atomic models or density maps or other things. You can use different models of image formation. You can use different ways of representing kind of poses of the molecules. How you decide to represent kind of orientation of molecules, et cetera. And it really enables diverse cryo-EM analyses, including, like we talked about before, localization of particles, determining molecular configurations, and even reconstructions. So that in cryojets, it's really, again, like to think about it, it's this way of kind of very flexible, very powerful way of modeling the image formation process, which then you can leverage all these kind of very fancy and powerful statistical analysis tools from Python and JAX. Okay, so to give a sense of what this enables, and this is kind of a toy example, just to give you like a sense of kind of possibilities. So before, I was talking about how like, okay, you can match a template. So you have like a ribosome structure, you try to, determine, you try to match it to data. And if you're doing that, you could think the parameters you're trying to optimize are the potential location and orientation of the template, right? So if there's like X, Y, and Z positions, that's 3 parameters. And then you have 3 Euler angles, so that's like 6 parameters. And so you can optimize over that. But instead of just optimizing location, one can actually think about optimizing the position of individual atoms within the template. And like I said, this is just kind of a toy illustration, but hopefully it shows the principle. So for example, and I should note this was done. This work was done by David, who's at Yale. And so what he did is that he said, okay, so he has some molecule with some real conformation, and he simulates a bunch of potential cryo-EM images from that molecule. And then he imagines that he has a template which is of the same molecule but has a different conformation. Right? And there's different ways you can talk about kind of representing, like the conformation of a protein, but here literally talking about the C alpha positions of all of the residues of this protein. And then you can see like, like if you kind of like, put this mismatched template on top of the true conformation, like they're close, but they have regions where they don't overlap. And then what he did is basically just, optimize over, allowed each of these 5,382 different C alpha, atomic positions to independently move around, and then optimized, what is their motion, optimize their position in order to kind of most overlap, by comparing to the cryo-EM images, and then try to improve the position of the atoms, and then he's able to show that he can actually get things to align, right? And so this is a bit of a toy problem because one wouldn't actually want to literally do stuff this way in practice. But I think it shows the principle that instead of just trying to optimize for a molecule, fits rigid body degrees of freedom, one could literally think about optimizing for thousands of degrees of freedom for each guy. And this is really enabled by using something like cryojets. All right. Okay. So then that's the developed tool. And now what Michael is doing is trying to apply this to understand microtubule systems. And this is very much a work in progress. And so I'll just give you a taste of what he's doing and kind of where he's going. All right, so what the basic idea is that, so one will have cryo-EM images of motors and microtubules in situ. This could be in vitro, this could be in cells. Then use this cryo-EM data analysis to uncover ensembles of the dynamic steady states of microtubules interacting by molecular motors, and then combine that with theory simulations and experimental microscopy to inform biophysical models. All right, so that's big picture goal. Okay, so the first step is really trying to think about this is like mapping out the lattice of individual microtubules in vitro. And so for, imagine this kind of, in some sense, it's a toy problem, but it's a problem which is very interesting in of itself. Imagine a microtubule, just single microtubule in vitro with motors walking on it, right? And then taking cryo-EM data and then asking first, where is each tubule and dimer in this image? All right, for every tubule and dimer, is there a motor bound to it or not? And then if so, if there's a motor bound, what conformation is that motor in? All right, and so the idea is to essentially, develop this kind of, posteriors of given certain kind of cryo-EM data, what's the probability that, what's the probability of a dimer being located there at a certain orientation? And then again, or given the, kind of cryo-EM data, what's the probability that there's a motor there? And what's the probability of that motor's conformation, right? So the idea is to kind of find those things. Like I said, work in progress. So first thing is just about trying to localize tubule and dimers using the known structure of microtubules as a template. And so the idea is that Evan Nagalis has done a lot of structural biology of microtubules, and we know high resolution structures from her and others. So talk a little bit about microtubule structure. Okay, so microtubules are made up of tubulin heterodimers, right? So a heterodimer has alpha tubulin and a beta tubulin subunit. The heterodimer is 8 nanometers long, so that's 4 nanometers between each subunit. And alpha and beta tubulin have very similar structures, but not literally the same. All right, and these tubulin heterodimers stacked to form protofilaments. All right, so this kind of stack is called a protofilament, and the protofilaments, arrange, join together to form a pseudohelical arrangement, which is a microtubule. All right, so that's structure. All right, so then here's this initial thing. All right, so here's a cryo-EM image of a microtubule, and then Michael Bryan uses kind of essentially template matching with cryojaps to try to localize this kind of template, which is this kind of cross section of a microtubule. And then what you see when he does this is that you see these 4 nanometer kind of very high, like significant repeats along the backbone of the microtubule. Right? Why 4 nanometers is because to some first approximation, or if alpha and beta were this, if you treat each subunit as really being different, there would be an 8 nanometer repeat. But because they look so similar, what you see is a 4 nanometer repeat. And then the idea is that you can just dock in this template and be like, oh, okay, here's how I think things are in this image, because I think I've identified these kind of template locations. All right. So, I mean, actually, I think this is kind of amazing that you can actually see this kind of 4 nanometer repeat when just looking at this image, the information's really there. Okay, but how well? Yeah.
Do you see defects through this method?
And so hold that thought. Okay, I'll come back to that later. So, but right now, that's not. But that's definitely something which we want to go towards. Okay, one question you can ask is how accurately can one expect to localize these tubulin dimers in cryo-EM images using this type of approach? And all right, so what I'm going to show, yeah.
Can I ask another question?
Yeah.
Are you also able to dock or match alpha and beta tubulins?
I'll come back.
Polarity?
Yeah, I'll come back to that in a moment too. I'll come back to that in a moment. Okay, but right now we're just going to ask just for these kind of peak localizations. And so this is just to get a sense of stuff. So what Michael did is that he simulates cryo-EM images of microtubules, and then in these simulated images where he knows what the ground truth is, attempts to localize tubulin dimers and compares with the results. Okay, so obviously this is going to depend on the signal to noise of the images. So if one assumed that one had a signal to noise of 1 and you simulate a cryo-EM image, it would be like this, right? And you get one thing. But if you simulate a cryo image with more realistic signal to noise, which is roughly 10 to the minus 2, and plot it on the same scale, it looks more like this, which maybe isn't super. I don't know. You wouldn't feel so hopeful if you stare at this. Nonetheless, okay, if you actually try to match stuff up and you measure localization precision as a function of signal to noise, okay, with signal to noise of 1, you can get insanely good values. But even with 10 to the minus 2, you can get a localization precision of roughly 0.1 angstroms with realistic signal to noise. Okay, 0.1 angstroms. Okay, so first when I was like, that's insane. That's not, okay. But if you think about it, in light microscopy, it's fairly typical to be able to localize a particle to within 1/10 of a pixel. You can do that without, crazy high signal to noise. All right. In cryo-EM, your pixels are an angstrom, right? And so localizing stuff to 1/10 of a pixel, not so ridiculous. All right. So I think this is actually again, this is just kind of simulation results, which we have to kind of confirm with more stuff. But I think this is probably realistic that you can localize stuff to within 1/10 of an angstrom. All right. Okay. So now microtubule polarity. All right. So microtubules are polar polymers. The two ends of the microtubule are not equivalent. You have a plus end and a minus end. And then you can ask how well can you determine polarity from these, just by looking at individual microtubules. And again, preliminary analysis of both experimental data, I'll say, but also simulated images, is that polarity can be defined with very, very high fidelity. We haven't quite quantified that yet, but likely, success rate is probably the error rate is probably less than 1%. And so I think this is not really a challenge. It's kind of very straightforward from cryo-EM data by this template matching, just because the structure is so different when you flip one way with the other. If you just literally rotate the microtubule around, right? Yeah, and I find it interesting because it's a very the cryo-EM analysis. It's a very different regime than we're used to in light microscopy, right? Because with light microscopy, at least when I think of fluorescent microscopy nowadays, I think of relatively high signal, but low resolution, so everything looks like a Gaussian blob. Here you have just insane resolution, but very low signal to noise. So it's a really different kind of way of thinking about stuff. All right. So then the next question is, can one determine seam position and alpha and beta subunits? Okay. So what do I mean by that for people who aren't familiar with this? Okay, so microtubules aren't a true helix. They're pseudo-helical. And so what I mean by that, okay, so if you look at kind of the connection between most protofilaments, it's really between them, it's like alpha is next to alpha, beta is next to beta. And that's true kind of most places in the microtubule, except for one particular place called the seam, which you have alpha next to beta. All right. Okay. And so then even if you know kind of the location of all the subunits and you know the polarity, you still need to determine where is the seam position. And then even if you know the seam position, even if you knew that the seam was here, you would still want to know who's alpha and who's beta. Okay. So let's just okay. Oh yeah, let's think about this. If you had 14 protofilaments, if you had microtubules with 14 protofilaments, there are 14 possible places the seam could be. And then given you have a seam at some location, there's kind of 2 possible registries of kind of who's alpha and who's beta. So you have 28 possible pseudosymmetries that you have to kind of compare. All right. And so if you were to try to think about this a little bit more, if you have a microtubule, which has the true seam at this location, and if you were to kind of misguess where the seam was by 2 protofilaments, essentially what you're doing is that you're misassigning who's alpha and beta for everyone in those 2 protofilaments. That's kind of what you're doing. And then, okay, so then you can kind of guess what you'd expect to see from that. So because you kind of have a constant amount of mismatch for each misaligned protofilament, this implies a sawtooth variation in the log posterior where the log posterior is the log posterior of kind of the correct pseudosymmetry given the image, where R is like a registry index which defines like which of these possible 28, and for the case of a 14 protofilament microtubule, 28 possible registers you have. And so the reason, because sorry, so like yeah, okay, awesome. All right, okay, so well, okay, I won't get into it, but anyways, that's what you expect. And then because we can investigate with simulated data. So again, simulate cryo-EM images of microtubules and attempt to assign seam positions in alpha beta to subunit identity. Okay, if you have a signal to noise of 1, you can, you really see the sawtooth pattern here. We didn't explore all possible registries, just kind of you can kind of do stuff to give you a guess of the decent guess of the right registry, but you can still see this kind of expected sawtooth pattern. And you can, it seems like one can assign the seam and the alpha beta densities with a very high fidelity, certainly with such crazy high signal to noise. But even with simulating cryo-EM images with realistic signal to noise, again, this is preliminary, but one does also still see a sawtooth pattern. And it does seem that one can still assign both the seam position and alpha and beta identity with quite high fidelities. If you're looking at this is log posterior, so it doesn't look like so much of a difference, but the best kind of hit registry, which is the true one, is still significantly better than anything else. All right, so then I'll finish up. So next step, okay, so we want to really rigorously test this tubular localization seam assignment and assignment of alpha and beta identities. And the idea is to do stuff similar to what I said Nico and Brownman have done, where we kind of, so for example, people, or Evan Nagalis has shown that the structure of microtubules at the seam is different than elsewhere, or the protofilaments is different. And then we can ask if we can kind of not put that into our template matching and then recapitulate, actually do a reconstruction of it. And that will show that the stuff is really working like we think it is. And so related to what was asked, right now, we're just using a rigid template. But what we've started doing is actually optimizing over lattice structures, right? So that can be a free parameter, the details of the lattice geometry, and one can have that space, do that in a spatially varying fashion. So really look for microtubule, look for defects and distortions in the microtubule and evaluate those. And so also like one hypothesis is that the GTP cap, the lattice structure is actually different than the bulk of the microtubules. One should, in theory, be able to observe that if it's right. And then ultimately, we want to go to motors and other maps on microtubules and be able to assign localization and conformations. All right. So like I said, okay. So this is the group. Thank you, everyone in the group. This is all everything I talked about was really Michael O'Brien, basically, or at least all the cryojacks and related stuff. And then, a lot of fantastic collaborators and other people doing stuff on cryojacks, and then generous support from Simons Foundation, and depending on the time, sometimes NIH or NSF. All right, thanks.
Or understanding of cell biology. And now, yeah, I think we have all the atomic details of all the molecules, and we also have all the biophysics experiment that we can get the dynamic of all the molecules, and how possible it is if we can integrate all the cryo-EM data of all the atomic level conformation information and dynamics we get from the biophysics experiment. We combine those to predict the dynamics of the cellular dynamics. Is that possible?
Well, yeah, no, that's definitely the goal, 100%. I mean, like I was saying, I think one of the challenges is that we know the atomic resolution stuff, we know the cellular stuff, but we don't really know how the atoms fit in at this kind of mesoscale. I think that's really totally unknown, and that you can't just get from solving structures, and you can't get that just from looking at stuff at the cellular scale or how stuff moves around, even doing single molecule tracking or something like that. I think we really need to know in this kind of mesoscale, how do things actually behave in context? And so I think one really needs, such approaches to really investigate stuff in context. And then I think that will allow us to create models that hopefully bridge these scales.
So Dan, thanks a lot for this lecture. Three related questions. First, you didn't really tell us what the objective function is of the template matching. Second is, do you solve it with gradient descent? Can you map it? And third, have you looked into the uniqueness? I mean, I completely believe the alpha beta tubulin story, but I think as soon as you guys go now into the motors, you might have actually non-unique solutions, right?
Yes.
A huge problem.
Yeah, no, 100%. Yeah, and so the gist of the objective function is that so basically you're kind of have a model of okay, so you write down a model of image formation, which is essentially a model of what the electron density is plus some noise. And then if you kind of whiten the noise, it becomes Gaussian. And so then it's essentially some like, and therefore the objective function is just like I'm going to screw it up. So it's either like the log or the Gaussian of the difference between the data.
Eventually.
Well, no. I mean, because it's really like a full Bayesian thing. I mean, like, and so it's really a full Bayesian thing, but it's really like that difference between the. So it's really the probability you generate the data given this kind of like model you have and this given this structure and the noise model, right? And so actually we have been using Gaussian noise, which is typically what people use in this, but there's no reason one is limited to that. If we want to use non-Gaussian thing, because we're doing everything computationally, we can. And like you said, yes, and so we're using gradient descent. So like a stochastic gradient descent approach, and one can map out the full posterior, basically, not just looking at like, so it's not just a maximum likelihood. You again can do like a full Bayesian thing. Yeah, so we haven't used gradient descent so far, but one could imagine using other approaches, which again is like, the way I would view this, it's really like a machinery to do all of that. So like the details of what approach you use might depend on your application, but it's just still like a machinery to allow one to do different things. Is how I would view it. And then the non-uniqueness is so that is, I would say, a huge issue, right? And so just because more generally, like the approach which I'm advocating here, which we're going after, is essentially you have some model which you're matching to your data. And then at the end of the day, what you're always saying is like, oh, is this model better than that model? But like, it could be that like, you just haven't chosen the right model at all, right? You know what I mean? And that's a very hard problem. And so that's why I think this question of validation is so important, of doing stuff like I was saying, like trying to use stuff like leaving out part of a template and using that to reconstruct what's there. And so then by using those type of things, even for like motors, one can try to do that. So for example, like if we have motors and that we assume and that we assign to different conformations, you should be able to actually reconstruct part of the motors and which come from different conformations and see different things and make sure that they vary in the right way. So you have some kind of internal control of that. And then of course in vitro, you can do other control experiments, like ask like, oh, here's the conformation I see if I have a lot of ATP. Here's the conformation I see if I have a non-hydrolyzable ATP, and just make sure you get the right things. But it's definitely a concern, and one has to keep on thinking about the right ways to deal with that. Yeah.